Leanne is a professor of psychiatry and behavioral sciences at Stanford University School of Medicine. Among her many roles at Stanford, she is a founding director of the Stanford Center for Precision Mental Health and Wellness, and of the Stanford Pan Lab for Precision Psychiatry and Translational Neuroscience. She is also the Associate Chair of Translational Neuroscience at the Department of Psychiatry and Behavioral Sciences. Prior to joining the Stanford community, Leanne was the founding chair of Cognitive Neuropsychiatry and directed the Brain Dynamics Center at Sydney Medical School. Leanne's center and translational programs integrate advanced neuroimaging, technology, and digital innovation to transform the way we detect mental disorders, tailor interventions, and promote wellness. She has developed the first taxonomy for depression and anxiety that quantifies brain circuits for diagnostic precision and prediction. Leanne has contributed over 300 scientific papers to the field and is the recipient of numerous awards, including the Young Investigator Award from the International Schizophrenia Congress and the Presidential Award from the American Psychosomatic Society. Today, Leanne will tell us more about the latest in precision mental health. Thank you, Leanne. It's an honor for us to have you here today at DECODE 2020. The virtual stage is yours.
Thank you so much, Kunal, for that generous introduction, and thank you to Inscopix for the opportunity to share this bench-to-bedside approach. I'm very excited to share how we're thinking about developing a precision medicine for mental health, and it's a concept that I refer to as biotypes. For years, we thought of cancer as the silent killer, but thanks to advances in precision medicine, we're able to detect subtypes of cancer earlier, and we're able to tailor treatments accordingly. Sadly, depression is now our number one silent killer, and that is especially true for 15 to 24-year-olds. Depression accounts for more diseases than cancer and many other diseases combined. Globally, the numbers are staggering. Globally, we have 1 million suicides per year. 10% of the population are affected. That's 615 million people with depression and associated anxiety. And we lose $1 trillion from our global economy due to the lost talent and lost productivity of depression affecting our young people. So it's not surprising that depression is now a number one cause of disability. What I'm excited to share with you is a solution and a path forward for these problems. We've known about these problems for a long time, and we need a radical new approach. The approach that I'm developing through my center is one I call biotypes. And in this approach, we think of distinct dysfunctions in large-scale brain circuits that create different subtypes of depression. And this approach to biotypes is possible because of advances in advanced imaging and a way to peer into the brain in action. And it's also possible because of advances in computation. And the research that I'll talk about draws on the Human Connectome project. We also target this in the same way that precision medicine for cancer did, at doing better for treatment in depression. And currently, only 3 in 10 people achieve remission on antidepressants after the first one they try. And we are on track to at least double that rate. And how we think of it is similar to what has been achieved in cancer medicine, whereby identifying subtypes through imaging and through genetics, we can do better with targeting the treatments. And similarly in cardiology, more recently through imaging and combining those data with lifestyle factors and other information, again, we can do better in personalizing the treatment. So how we do this is in developing a roadmap for this radical new approach to biotyping, and it occurs in 5 steps. The first one is to use the advanced imaging to identify the biotypes. And in this case, we're looking at least 6 different types, biotypes or subtypes of how these large-scale brain circuits are disrupted. By no means do we think that imaging is the only source of information we need to consider, but we do anchor the biotypes in brain circuits as the most proximal measure. And how you could think of an analogy is when we're thinking about heart disease and hypertension, the organ of interest is the heart. So in mental health, the organ of interest is the brain. And these large-scale circuits are the ways that regions within the brain communicate in an organized way with each other. And I'll touch on this in a moment. We integrate these data with other sources of information, so self-reported symptoms, genetics, physiology, and behavior. And then we take at step 2 those biotypes into mechanistic trials. And the goal there is to ask, do treatments not only improve symptoms, but do they improve these biotypes? And we need to know that in order to do better about understanding how do the treatments work and for whom. Once we have that information, we're very excited about new approaches to prospective trials, which are biotype guided. And in this case, we identify the patient based on their biotype. And we look at novel therapeutics that are specifically selected to target that biotype. Of course, all of this creates a lot of data, so we rely heavily on advances in computational models and data-driven approaches to integrate these data. What that also makes possible is the opportunity to scale and to be able to build on the biotypes with sensors and wearable technologies. And very importantly, we want to accelerate the clinical translation of this approach. And recently, we have launched a clinic to make this possible, and I will come back to that as well. The data I'll show you come primarily from 3 large trials funded by NIH. One of them was called the RAD project, where we assessed 160 people for depression, anxiety, and we identified their biotypes. And this was possible because of an excellent team of postdocs and graduate students and coordinators. The other one is an international trial called ISPOTD, which I led across 5 different countries. And here we biotype people with depression before they started treatment. Then we assigned them to treatment and reassessed their biotypes and symptoms. In the third trial, this was a partnership with my co-PI, who's now at UIC, June Ma, called the Engage study. And in this case, we were looking at a behavior therapy focused on problem solving. And we assessed biotypes, again, prior to being assigned treatment, during the treatment, and then after the treatment. And this, as I said, was a behavioral therapy. So let's have a look at 4 of these biotypes. The first one, the blue one, number 1, it involves the default mode. The default mode occurs when the brain is at rest, self-reflecting. When it's disrupted, it's over-connected and we can worry and focus too much on our internal thoughts, which can be called rumination. In the case of treatment, those people with intact default mode tend to remit on standard antidepressants, but as shown in yellow, when this default mode is disrupted, we have a high accuracy cross-validated of predicting those who do not remit. The question, of course, is what may they do well on? And what we have found in preliminary trials is those who do not remit on the antidepressants because of their biotype do tend to do well on transcranial magnetic stimulation, a new treatment recently approved. If we look at the negative affect circuit, this is biotype 3, it is engaged when we focus on negative emotions, including facial expressions, and it can be overactive when it's disrupted and create an over-attention or a negative bias, and that disrupts our ability to focus on positive. What does this mean for treatment? What we have found is that those people who respond to standard SSRI antidepressants tend to have a flattened negative affect circuit, but the biotype here, biotype 3, with negative bias, these people tend not to respond. So again, the question is, what might they respond to? And what we have found in our ENGAGE trial is this biotype does respond to the problem-solving therapy, the behavioral therapy. The fourth one, which is number 6 biotype, involves the cognitive control circuit, which helps us with our planning and executive function. It is engaged when we try to inhibit, say inhibit red and press green, and the frontal brain is disconnected. It can cause a cognitive fog and problems in concentrating. What does this mean for treatment? What we find is a very distinctive pattern with this biotype. So we find in this case the under-connection and under-activity of this cognitive control biotype does not respond to standard SSRIs. However, it does respond to more atypical SSRIs that have a multiple effect on multiple neurotransmitters. And we are following up on this in a new trial, which is a biomarker-guided study. We're looking at a more targeted novel therapy for depression, which has not previously been used. It's called guanfacine, and it targets this biotype involving the dorsolateral prefrontal cortex, which we need to control cognition and concentration. The fourth biotype involves the positive affect circuit or the reward circuit. And what happens is when that is disrupted, we tend to get a flattening of the reward centers, which are known as the striatum, or ventral striatum in particular. And it causes a concept of anhedonia, a blunting of emotion and a flattening, and everything feels gray. We are excited to have piloted a new treatment called pramipexole, which targets the striatum and is not typically being used for depression. What we have found is in our first 5 patients with this biotype, they show on pramipexole an improvement in the biotype, an increase in the ventral striatum, and they show an improvement or a decline in the anhedonia symptoms. And this gives us promise for this biomarker-guided approach. One of the findings, the one I'm highlighting, is the story of Joe, who when he came to us was unable to work. He was very suicidal, very distressed, and he shared his story in the Science magazine. And he, now 9 months later, is doing well and able to work again. So turning briefly to our current times, let's think about what biotypes might mean in understanding the impact of the pandemic. We have published 2 papers so far on really taking a look at the biotypes in terms of how might they give us a way to understand how a chronic stress, such as the pandemic, may be disrupting all of our brain circuits. And how does that help us think about tips, behavioral tips? So here we've thought about experiences of rumination, even if not in the clinical form, may mean that behavioral tips could work. Scheduling worry time, limit exposure to sources of worry, for example. And similarly, for the cognitive control brain type that causes cognitive fog, here it's a recognition that we're all going through massive brain plasticity. So we need rest time for our brain plasticity to catch up. We're also excited that we've launched a study to look at how recovery from COVID impacts biotypes. So for those who've been unfortunately affected, how are they recovering 3, 6, and 12 months later? And can we help in understand the effects on the brain circuits and on symptoms? So to end, I want to envision how we could think about this biotype approach for the future and how do we accelerate clinical translation. I'm excited that we've launched a new clinic. It's called the Stanford Translational Precision Mental Health Clinic. One of the challenges has been not only to develop the scientific insights, but to create a new workflow, one that doesn't exist for mental health. So how do we envision a flagship center that could have all of these new technologies available, all of the new therapies, ways to test to personalize therapies that we don't currently have available for clinicians? And how do we then think of it in a beautiful and uplifting setting? And how do we use that flagship to ultimately scale the findings so that they can be used by many more people and made more available? We've been beta testing this approach, so we've looked at how could we use digital platforms to share the findings from not only the biotyping, but from the symptom measures and the behavioral measures. We've also thought about if we establish this biotyping platform and it's robust and reliable as we are seeing, we then have a means to use it to anchor how we scale and how we link biotypes to information from mobile devices and from wearable sensors. And in that sense, we are thinking about how transformations have occurred in other areas of medicine that are complex and stigmatized. We can think, for example, of the revolution in heart health that started in 1948, so only 72 years prior to technology. We went from not knowing anything about the heart to having imaging technologies, to having an understanding of what creates risk. What do we have to do to prevent heart disease? And now we have heart health apps and ways to track our risk. And this is what we need now for mental health. So my call to action is let's think about how to scale from neuroscience to the real world and to the field, and do a cohort study that is similar to making the transformations that occurred for heart health and for cancer medicine. Thank you very much, and I look forward to your questions.
Thank you, Leanne. Fascinating talk to kick off this second segment with respect to the potential for diagnostics and therapeutics at the bedside. So a few questions have come in. The first is kind of taking advantage of the ML AI techniques that have obviously dominated several fields, including medicine. And the question is, as AI improves further, how do you see that impacting perhaps the subtype profiling?
It's a great question. So how I see it is we have an explosion of data that we're gathering, clearly. And what I've focused on is a reproducible way to quantify the biotypes, similarly to how you would think about, say, an EKG. You want to know that it's robust and reliable and can be used in the clinic. What I envision with AI is it will give us the insights that we currently don't have the capacity to see in the data with our traditional approaches. So we would want to look at how do the biotypes interact, how are they over time, what do they predict over time, and use ultimately AI algorithms to help us understand individual profiles of biotypes in real time.
And I guess a related question which came in, which just literally picks up on what you're alluding to, is what do you see as the challenges or maybe the opportunities to look at it from a different perspective for scaling up some of these techniques and ultimately, getting these fMRI biotype-based diagnostic markers into the clinic?
Right. The opportunities are there, so I'm very encouraged by the collaboration I have with neuroradiology, who would say if the reproducible biotype procedure is there, then their job is to make it scalable. In the same way that we wouldn't think twice about getting a scan for a broken leg, and we wouldn't think twice about getting a scan if we went to neurology. Of course, all the cost models need to be worked out, but I believe it's feasible. And my personal view would be if we have that anchor in understanding the brain, we have a platform to then be able to understand more scalable, more distal measures. And those might be app-based, or they may be wearable, or they may be able to be done remotely, but we can interpret them relative to how we understand the brain. And that gives us an objective, measurable way to understand the impacts of all other information. And that includes, of course, lifestyle factors as well and life history.
Mm-hmm. No, that's certainly extremely inspiring if we can get some of these techniques as part of the standard of care in the future, just becoming a lot more objective about diagnoses and getting diagnostic capabilities as part of standard of care. Certainly very appealing vision. One last question in the interest of time, very practical question actually. How can people enroll for some of these clinical trials that you're running?
We welcome people enrolling, of course. So on the link on the slide that's being showed now, the URL, I should say, that lists the studies that we have available, and it has links to actually expressing interest in the studies. We also have a resource page on that website that explains the different studies and resources and will provide updates on when new studies resume and also when our new translational clinic opens, which is on schedule for next year.
Terrific. Well, thank you so much, Leanne. Really enjoyed the talk, and what a way to kick off the second segment of the DECODE Summit, focusing on diagnostics and interventions that are at the bedside and are resources for folks today.